Hello everyone, welcome to my channel Life Science Gyan. In this video we will see question paper of Kurukshetra University Assistant Professor exam of Microbiology with solution. Our first question is the tiny microorganism observed by Leeuwenhoek's wide-ranging investigation was called as animalcules, bacteria, fungi, moving animal. So the correct option is option 1, animalcule. Our next question is which one of the following was among the first to test spontaneous generation theory? Francisco Redi, Leeuwenhoek, Dennis, Parker. Correct option is option 1. Francisco Redi was among the first to test the spontaneous generation theory of evolution. Our next question is which will come under the 5 eyes of culturing microorganisms? Identification, incubation, isolation, all of the given. Correct answer is all of the given come under the 5 eyes of culturing microbes. Our next question is reducing media is essential in determining which one of the following requirement? Oxygen, iron, carbon dioxide, none of the given. Correct option is option 1, oxygen. Reducing media is essential in determining oxygen requirement. The next question is which one of the following is the capacity of some microbes to modify their biological processes, reproductive strategies, or morphology in response to environmental factors? Ceopodium, pleomorphism, sarcinophore, tetrad. The correct option is option 2. Pleomorphism is the capacity of some microbes to modify their biological processes, reproductive strategies, or morphology in response to environmental factors. Our next question is the algae that can grow equally well as phototrophs and as heterotrophs are called as facultative mixotrophic algae, obligate phototrophic algae, obligate heterotrophic algae, obligate mixotrophic algae. The correct answer is option 1. The algae that can grow equally well as phototroph and as heterotroph are called as facultative mixotrophic algae. Our next question is the bacteria which move toward an attractant is called as positive chemotaxis, negative chemotaxis, inhibition, none of the given. So correct option is option 1. The bacteria which move toward an attractant is called as positive chemotaxis movement. The next question is if an organism approximately 9% or more sodium chloride for growth, it is referred to as extreme halophile, moderate halophile, halophile, none of the given. So correct answer is option 1, extreme halophile. If an organism can tolerate 9% or more NaCl for growth, then it is known as extreme halophile. Our next question is the direct genetic examination of the genome present in an environmental sample is known as genomics, metagenomics, transcriptomics, gene silencing. The correct answer is metagenomics. In metagenomics, direct genetic examination of genomes is examined. Our next question is the enteric bacteria Staphylococcus aureus will develop drug resistant to which one of the following by developing resistant to biochemical pathway? Chloramphenicol, sulfonamides, streptomycin, erythromycin. The correct answer is sulfonamides. Our next question is, which one of the following compound present on the enzyme small non-protein molecules that participate in catalysis but are not themselves substrate? Prosthetic group, coenzymes, niacin, none of the given. So correct answer is option 1. Prosthetic group of compounds present on the enzyme small non-protein molecules that participate in catalysis but are not themselves substrate. The next question is, the characteristic of catabolite repression that causes two phases of growth is called as bidirectional, dioxic, exponential, none of the given. So correct answer is option 2, dioxic growth. This is the characteristic of catabolic repression that causes two phases of growth. Our next question is, an energy poor and highly oxidized form of carbon, CO2, is reduced and incorporated into cell material through a process known as following pathway: heterotrophic, autotrophic, chemolithotropic, iron oxidative. So correct answer is option 2, autotrophic. In autotrophic pathway, an energy poor and highly oxidized form of carbon, CO2, is reduced and incorporated into cell material. Our next question is, which one of the following pathway is a variation of the glycolytic process, is commonly present in bacteria, particularly in species of pseudomonad group? Mixed acid, ED, PPP, and gluconeogenesis. So the correct answer is AD. The next question is, constitutional isomers include dihydroxyacetone and which one of the following, because they have the same chemical formula but different atom arrangements? Glyceraldehydes, aldose, anomer, mannose. So correct answer is glyceraldehydes. And dihydroxyacetones are isomers which are having same chemical formula but different atom arrangements. The next question is, lipids must possess hydrophobic characteristics in order to form DNA, RNA, membranes, protein. The correct answer is membranes. So lipids must possess hydrophobic characteristic in order to form membrane structure. The next question is, DNA's three-dimensional structure was first revealed in 1953, 1940, 1930, none of the given. The correct answer is option 1, 1953. The next question is, DNA fibers were photographed using X-ray diffraction by which one of the following scientists and Maurice Wilkins? Pasteur, Rosalind Franklin, Francis Crick, Watson. The correct answer is option 2. DNA fiber photograph by using X-ray diffraction was produced by Rosalind Franklin and Wilkins. The next question is, molecules of small nuclear RNA, snRNA, take role in splicing of which one of the following exons? RNA, DNA, mRNA, none of the given. So correct answer is option 1. snRNA take role in the splicing of RNA exons. The next question is, DNA polymerases which encourage the creation of the bonds connecting the units of the DNA backbone were identified by which one of the following and his colleagues from E. coli? Crick, Arthur Kornberg, Franklin, Watson. The correct answer is Arthur Kornberg identifies DNA polymerases which encourage the creation of bonds connecting the unit of the DNA backbone. Our next question is, which virus performs replication of genome in cytoplasm? Herpes viruses, orthomyxoviruses, pox viruses, none of the given. So correct answer is option 3. Pox viruses perform replication of genome in cytoplasm. Our next question is, which of the following is or are an example of derived lipids? Carotenoids, terpenes, steroids, all of the given. So correct answer is all of the given. All of the given are example of derived lipids. The next question is, in bacterial growth curve, which phase represents the initial adjustment period? Log phase, lag phase, stationary phase, death phase. The correct answer is option 2. Lag phase represents the initial adjustment period in bacterial growth curve. Next question is, central dogma was proposed by Altman, Robert Hooke, Franklin, Francis Crick. The correct answer is Francis Crick. Our next question is, what is the rate determining and rate limiting step of Michaelis-Menten kinetics respectively? The product formation step and the complex formation step, the complex dissociation step to produce products, and the product formation step, the complex formation step and the complex dissociation step to produce products, none of the given. So correct answer is option 2. The complex dissociation step to produce products and the product formation step is the rate limiting steps in Michaelis-Menten kinetics of enzyme. Our next question is, during bacterial growth, secondary metabolites can be produced in which phase of growth? Lag phase, log phase, stationary phase, death phase. So correct answer is option 3. Stationary phase is the phase where secondary metabolites can be produced by bacteria. The next question is, the enzyme involved in the citric acid cycle are present in inner mitochondrial membrane, cytoplasm, mitochondrial matrix, plasma membrane. So the correct answer is option 3. Mitochondrial matrix, in which citric acid cycle enzymes are present. The next question is, what is the diameter of the smallest cells of mycoplasma? 1 meter, 0.3 micrometer, 0.5 micrometer, 0.1 micrometer. So the correct answer is option 2. 0.3 micrometer is the diameter of smallest cells of mycoplasma. Our next question is, how many steps are involved in the translation process? 2, 3, 5, 4. The correct answer is option 4. 4 steps are involved in the translation process. Our next question is, name the enzyme that differs in amino acid sequence but catalyzes the same chemical reaction. Isozyme, abzyme, ribozyme, allosteric enzyme. The correct option is isozyme is the name of enzyme that differ in amino acid sequence but catalyzes the same chemical reaction. Our next question is, which one of the following plays a crucial role in cell signaling as they can detect chemical signals or physical stimuli? Pili, flagella, receptor, all of the given. So correct answer is option 3. Receptor plays a crucial role in cell signaling as they can detect chemical signals or physical stimuli. The next question is, macrophage present in the liver is monocyte, Kupffer cells, sinus, histiocytes, alveolar. The correct answer is option 2. Kupffer cells are macrophage present in the liver. Our next question is physical, physiological, cellular and cytokine barriers comes under adaptive immunity, passive immunity, active immunity, innate immunity. So the correct answer is option 4, innate immunity. So physical, physiological, cellular and cytokine barrier comes under innate immunity. Our next question is P53 is located on which one of the following and activated by which one of the following respectively. Option 1, 17th chromosome phosphorylation, 16th chromosome dephosphorylation, 15th chromosome methylation, 14th chromosome and carboxylation. So correct answer is option 1. P53 is located on 17th chromosomes and activated by phosphorylation. The next question is the experiment confirms that bacteria have the ability to transfer genetic information through the following process: translation, transformation, transduction, transversion. So correct answer is option 2. Transformation is the process which confirm that bacteria have the ability to transfer genetic info through the transformation. The next question is the role of beta-mercaptoethanol in SDS page: polymerization, oxidizing agent, separation, reducing agent. The correct answer is in SDS page, beta-mercaptoethanol is a reducing agent of protein. Next question is in the living cells, the oxidized metabolite is reduced into which one of the following form, particularly in the absence of an inorganic electron acceptor: lactate, oxygen, sodium, water. The correct answer is option 1, lactate. In the living cells, the oxidized metabolite is reduced into lactate form, particularly in the absence of inorganic electron acceptor. Our next question is the bacteria which involved to degrade the fermented product is methanogenic bacteria, sulfate reducing bacteria, carbon degrading bacteria, hydrogen producing bacteria. So the correct answer is option 1, methanogenic bacteria which involved in degrading fermented products. Our next question is the protein will precipitate in following pH: 1 to 4, 5 to 7, 7 to 9, won't precipitate at any pH. So the correct answer is option 1, at pH 1 to 4 protein will precipitate. Our next question is which is used in biodegradation of xenobiotics: enzyme, chemical, physical sorting, pH. So the correct answer is option 1, enzyme is used in biodegradation of xenobiotics. Our next question is the theory of nuclear magnetic resonance NMR is based on energy of the nucleus electrical moment, magnetic field is produced by the spin of nuclear electrical moment. So the correct answer is option 3. In NMR, magnetic field is produced by the spin of nucleus. Our next question, name the radioisotope used in molecular biology and genetic research: phosphorus 33, thallium 201, iodine 131, cobalt 60. So the correct answer is option 1. Phosphorus 33 is used in molecular biology and genetic research. Our next question is, which one of the following is the most effective method used to study the integral membrane of proteins? Fluorescence recovery after photobleaching, FRAP, cryosectioning and electron microscopy, freeze etching, freeze fracture technique. The correct answer is freeze fracture technique is most effective method to study the integral proteins of the membrane. Our next question is, H and L in the plaquette per month design denote height and length, high and low, half and life, hearing and listening. The correct option is option 2, high and low. So H and L in plaquette per month design denote high and low. Our next question is, the sparger made of metal or ceramic is combined sparger and agitator, porous sparger, nozzle sparger, orifice sparger. So correct answer is option 2, porous sparger is made of metal or ceramic. Our next question is, which one acid is not produced by Aspergillus niger? Oxalic acid, lactic acid, citric acid, gallic acid. So the correct answer is option 4. Gallic acid is not produced by Aspergillus niger. Our next question is, in quid pro quo stand for exclusive right to produce, market and use the innovation, none of the given, info made available to the public for the duration of the patent, monopoly is granted. The correct answer is option 4. In quid pro quo stand for the duration of patent, monopoly is granted. The next question is, which of the following products is acidic and has no aroma? Acidophilus milk, cultured buttermilk, Bulgarian milk, cultured sour cream. The correct answer is option 3, Bulgarian milk. The next question is, what is the range of the protein content in yeast cells? 40 to 50%, 20 to 40%, 69%, 12 to 15%. So the correct answer is option 1. So 40 to 50% protein content is found in yeast cells. Our next and last question, botulism can be prevented by adding chemical preservatives, proper low temperature before food canning, proper sterilization before food canning, none of the given. The correct answer is option 3. So botulism can be prevented by proper sterilization before food canning. Thank you. Please subscribe my channel, like and share the video. Thank you.